Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. So today I'm going to present a poster entitled Detoxification Mechanism in mouse bone marrow cells and multi-lineage hematopoietic stem and progenitor cell induced by 1,4-benzoquinone. Okay, benzene is known as a carcinogen that able to induce leukemia, and humans are exposed to benzene through occupational exposure or through environmental exposure. There are a number of benzene metabolites that are known to be toxic to the human body, and one of the hematotoxicity one of the metabolite that is known to cause hematotoxicity effects is 1,4-benzoquinone, or known as 1,4-BQ. And 1,4-BQ is a highly reactive benzene metabolite that can bind to cellular macromolecules such as protein and DNA, causing cellular damage as well as malignant transformation. Yeah, so that's why in this study, we want to investigate what are the detoxification mechanism that trigger or that driven the benzene toxicity in mouse bone marrow or in mouse hematopoietic stem cell niche, targeting different progenitors comprised of myeloid, erythroid, as well as pre-B lymphoid. Yeah. So as for the methodology, in this study we are using bone marrow cell, freshly isolated from mouse and cultured for 24 hours prior to 1,4-BQ exposure. And then the cells were exposed to 1,4-BQ exposure for 24 hours, followed by the bone marrow cells were harvested and divided into two groups. Okay, one group for direct detoxification study, and the other one for colony forming assay to get or to obtain the progenitor for erythroid, myeloid, as well as lymphoid. Yeah. And for the CFU assay, cells were cultured, or the CFU assay were cultured for 7 to 14 days, depending on the types of progenitors. And after that, the colony that are formed on the CFU assay were harvested and isolated for protein and analyzed for the expression of two proteins. One is detoxification proteins, namely NQO1, and the other one is a metabolism protein known as myeloperoxidase, or MPO. Okay, as for result and discussion, we noted that NQO1 expression was significantly reduced in myeloid and pre-B lymphoid progenitor starting at lower 1,4-BQ exposure. And then result also showed that myeloid progenitor was the most significantly affected as compared to other myeloid as well as lymphoid progenitors. In contrast, for MPO, we noted that for the MPO protein expression, there was an increase in MPO expression, especially in myeloid as well as pre-B lymphoid progenitor as compared to erythroid, when of which we see there is no significant effect on the erythroid progenitor. So as a conclusion, we noted that there is a reduced expression of NQO1 along with induced expression of MPO could be the detoxification mechanism mediated by 1,4-BQ exposure, with myeloid progenitor was found to be mostly affected as compared to erythroid and lymphoid progenitor. Yeah, so that's all from me, and thank you very much.